Good day, everyone. You're welcome to my YouTube channel, Everything Medical. In today's class, I'll be treating Cori cycle and Rapoport Leubring cycle under metabolism. Rapoport Leubring cycle can be called the 2,3 ppg shunt, which is phosphoglycerate. Now, before we delve into Cori cycle, let's look into the concept. What is Cori cycle? Cori cycle is a process by which glucose is converted to lactate in the muscle, and in the liver, lactate is converted back to glucose. That is the concept of Cori cycle. The essence of Cori cycle is to relieve the accumulated lactate in the muscle, which is clinically seen as a muscle cramp or muscle fatigue experienced by an athlete or a footballer during strenuous exercise. So during strenuous exercise, there is an accumulation of lactate or lactic acid in the muscle, which results clinically as a muscle cramp or a muscle fatigue. Now, let's dive into the cycle. We have here, you have the muscle, you have the blood, you have the liver. Cori cycle involves three significant organs. We have the muscle, the blood, and the liver. Now, let's check how the lactate diffuses to the liver through the portal vessel to form glucose through gluconeogenesis. And after forming glucose in the liver, it will then diffuse to the blood back to enter the muscle and supply energy to the muscle. By the virtue of doing that, the muscle are being removed by that accumulated lactate, which leads to muscle cramp or muscle fatigue during strenuous exercise experienced by an athlete or a footballer. Now, let's go. In the muscle, lactate diffuses from the muscle to the what? Blood. And in the blood, it gets to the liver through the portal vessel. In the liver, the lactate is oxidized to pyruvate. Pyruvate will undergo gluconeogenesis, or it will be channeled to gluconeogenesis to form glucose. Glucose is being generated. Now, once glucose is generated, it will diffuse back to the blood. From the blood, it goes to the muscle. Now, it forms pyruvate again, while pyruvate forms lactate. So that is how the cycle goes. This cycle works during anaerobic glycolysis, because in anaerobic glycolysis, pyruvate is converted to lactate. So it works when there is no presence of oxygen to oxidize what? Glucose. So that is it. So let's delve into the significance of Cori cycle. Significance of Cori cycle. Before I dive into the significance of Cori cycle, let's give the conversions their names of the pathway. Here, for pyruvate to be converted to glucose, it is a pathway known as gluconeogenesis. So let's write gluconeogenesis. Gluconeogenesis. While for glucose to be converted to pyruvate, it is glycolysis, or EM pathway. EM pathway. So, what is the number one significance of Cori cycle? Number one is that the process requires ATP. It requires ATP. That is the number one significance of Cori cycle. It requires ATP. Number two, the lactate produced by the muscle is efficiently utilized by the body. I repeat, the lactate produced by the muscle is efficiently reutilized by the body. That is the number 2 significance of Cori cycle. We go to number 3. In number 3, during serious exercise, the lactate stored in the liver is used to produce glucose. That is the number 3 significance of Cori cycle. In number 4, the number 4 significance of Cori cycle is talking about the oxygen depth. The what? The oxygen depth. Before I will explain oxygen depth, I'm going to be telling you the number 4 significance of Cori cycle. Number 4 is that the resultant increase in oxygen consumption is the explanation of oxygen depth after a vigorous exercise. Then what is oxygen depth? Oxygen depth is the oxygen that is needed to oxidize lactic acid to pyruvic acid, from pyruvic acid to carbon dioxide, to water, and chemical energy, which is ATP. So that is the concept of oxygen depth. Now you can flash back to the number 4 significance of Cori cycle, that it says the resultant increase in oxygen consumption is the explanation of oxygen depth after a vigorous exercise. So that is it. Now we have done justice to the Cori cycle. Let's delve into the Rapoport Leubring cycle, which can be called the 2,3-BP shunt. The 2,3-BP shunt. The 2,3-BP shunt. What is 2,3-BP shunt? Now let's look into a little bit concept of 2,3-BP shunt. Recall that in RBC, it does not have mitochondria. It lacks mitochondria. That is why glycolysis is the only pathway that sustains energy for the red blood cell, which is the erythrocyte. Without the energy from glycolysis, there wouldn't have been any energy supply to the red blood cell because it lacks mitochondria, which is the powerhouse of a cell. So now, the step 6 of glycolysis in RBC, it is bypassed. Automatically, the step 6 of glycolysis is always bypassed in RBC. That is why it had its own shunt in glycolysis. So other pathways in glycolysis occurs in RBC except the step 6. On reaching to the step 6, it will bypass to enter the step 7. That is why it had its own shunt called the Rapoport Leubring cycle, or the 2,3-BP shunt. So let's delve into the pathways. Here we have the glyceraldehyde 3-phosphate, which will give us 1,3-BP shunt, 3-phosphoglycerate, and finally to 2-phosphoglycerates. So that is it. Now here it goes this way. 2,3-BPG. It goes this way again. Now, as you can see, this is step 5 in glycolysis. This is step 6 of glycolysis. Step 6 of glycolysis. Why this is step 7 of glycolysis? You can see here. So in Panner's pathway, which is glycolysis, in RBC, it is always being bypassed in step 6. The step 6 of glycolysis is never found in RBC. Imagine, look at the pathways of glycolysis in RBC. So as it is occurring in RBC, on reaching to the step 6, it will bypass and form 2,3-BPG, continuing to BPG. So it will bypass it. Do you get? It will not form it. So it will do what? Bypass it and continues to form the step 7, because here a hole is the step 6. So now let's write the enzymes. Glyceride 3-phosphate is being catalyzed by an enzyme called glyceride 3-phosphate dehydrogenase. Why? For 1,3-BPG to form 2,3-BPG, it is being catalyzed by an enzyme called BPG mutase. BPG mutase is a mutase enzyme that does it. Now, for 2,3-BPG to be converted to 3-phosphoglycerate by removing one phosphate group, is being catalyzed by an enzyme called BPG phosphatase. That is the enzyme that converts 2,3-BPG to 3-BPG. Now, in the step 6, it is being catalyzed by a kinase enzyme. Because that ATP is being formed by a kinase enzyme, which is 1,3-phosphoglycerate kinase. Is the enzyme that converts 1,3-BPG to 3-BPG. That is the step 6 of glycolysis. Then recall that for 2,3-BPG to form 3-BPG, there is always a removal of one phosphate group. So we remove one phosphate group. So that is it. So this is how the reaction takes place. Recall that we have established earlier that in RBC, this whole step 6 is bypassed. So what that is being formed is 2,3-BPG. Instead of forming the step 6 of the glycolysis, it will bypass it and continues with the step 7. So that is how it goes. Now we have the significance of Cori cycle. The significance of 2,3-BPG or the Rapoport Leubring cycle. Number 1 significance. Significance of 2,3-BPG. The main significance is that this whole process does not require ATP because it lacks mitochondria. That is number 1 significance of 2,3-BPG. It lacks mitochondria. That is why it does not require ATP. Number 2 significance of 2,3-BPG. 2,3-BPG increases in fetal circulation. That is the number 2 significance of 2,3-BPG. It implies that it increases in fetal circulation. That's the number 2 significance of it. In number 3, 2,3-BPG binds with hemoglobin and reduces the affinity of oxygen towards hemoglobin. So in the presence of 2,3-BPG, oxyhemoglobin will unload more oxygen easily to the tissue. That is the number 3 significance of 2,3-BPG short. We go to the number 4. In number 4, the concentration of 2,3-BPG in red blood cell under hypoxic condition or hypoxia increases, thereby favoring the release of oxygen easily to the tissue, even when the partial pressure of oxygen is low. That is the number 4 significance of 2,3-BPG shunt. It's talking about the concentric increase in 2,3-BPG in higher altitude or high altitude favors oxygen dissociation. So that is all about the Cori cycle and the Rapoport Leubring cycle. So stay tuned for my next class.